So right now I'm just going to show you HiSat2. Once you have HiSat2 open, what you are going to do is, so this is what comes up. And you see, use a built-in genome, select a reference genome. So you can go through here and find the Homo sapiens genome, but our reference genome is actually the files that we downloaded, so the microRNA files. So we're going to go up here where it says use a built-in genome, and we're going to select use a genome from history, and from there we'll be able to choose our file. Now you're going to use the fused file, but I haven't made it yet, so for now we're going to deal with the UCSC file. So if you open the pull down, you find what file you want. This one is at the top because we recently uploaded it. I feel like I've said this on the other video where my phone died. Anyway, yeah, just select the reference genome you want to use. And then, is this a single or paired library? I'll have to explain what that means, but for right now, single end, paired end dataset collection, paired end data from single interleaved dataset. I'll let you know what type we're working with, but for now I'm going to click paired end data from single interleaved dataset, because I think that's what this one is. And then this is one of our subjects. This is must be data type FASTQ Sanger or FASTA. Pretty much after you're done with the quality trimming and quality score, throwing out the bad stuff, you're gonna work with FASTA because they're just smaller file types and you no longer need the quality information. For most of the stuff I do, that's true. You may run into situations where it's not true, like if you need all of the data possible and you have to end up working with low quality sequences, but you want to penalize them when you're mapping or doing whatever, like K-mer search or BLAST, whatever you're doing, then you might work with FASTQ files, but for now we're just going to work with FASTA files, which is just the sequence and the name. Specify strand information, unstranded. These, in my experience, I use them more with like prokaryotes, but just do unstranded for now. I can't like say that's the only time you'll ever use that. And then paired end options, you always want to look at all of your options and read everything on whatever software you're using. So paired end options use default values or specify paired end parameters. We will look and see what that is. So see alignment options below for more information. Select the upstream, downstream mate orientations or a valid paired end alignment against the forward reference. So it looks like the default is forward reverse, which is likely what we have, or actually it is what we have, I know that. So we're gonna go ahead and leave that alone, and everything else, you can read all of it, but it's not gonna apply here. So I'm going to go up and use the default values. Then summary options, open this. Output alignment summary in a more machine-friendly style. Because this is the first time we're running it, I'm going to click yes, print alignment summary to a file. I always want that. I always keep that for my records so I can see if there's an outlier subject, like a subject that only aligns or maps 70% and all the others map 85%. Maybe there's something wrong with that subject, the data from that subject, and I would want to look into that if it's skewing the results. So anytime there's a summary, you always want to click yes and then save that information in a spreadsheet. Oh, that's okay. We're going to click yes on both of those and see what happens. So under advanced options, we have all of this use default values, but we need to know what it is. So we don't really know what parameters we want to set for the mapping because we're figuring it out. So we're just gonna go down and read through. Input options. So specify input options so we can read it. Skip the first in reads or pairs in the input. We don't want to do that. Line the first in reads or read pairs from the input. We don't want to do that. We don't want to trim. Our data is already prepped, so we don't want to do any of that. Select quality score and coding. We don't have quality scores because we're working with FASTAs, so we don't want that. And then the rest of this is quality score and quality value stuff, which we don't have in our file. So we can feel pretty confident using selecting use default values. So next, alignment options. So this is how we deal with ambiguous characters. We don't have any ambiguous characters in our file. Ambiguous characters would be like this list. So sometimes if you don't know, it's usually files that you've worked with for something else. It's not off-machine files. Off-machine files will have A, C, G, T, and sometimes they'll have some N. I already know we don't have a lot of N because we selected to not have a lot of N's. But these are ambiguous characters, all of these. So if you have a W in a sequence, you might have an A there, or you might have a T there. We don't have those. So this doesn't really matter, but we're gonna go ahead and leave it as the default. Reads exceeding the ceiling. So this constant term, if you don't know what these mean, which I don't, I'm guessing it's dealing with ambiguous, but you would go on and you thought it was important, then you would go on to read more about it. And that would be in the README file for this program. So this program can be run through command line, which is pretty much what Galaxy is. It just puts all of this in the menu based. You can find it in the README. Sometimes Galaxy will have the README at the bottom, which it does here. So you can go and find whatever information you need to inform your decision of whether to use something. And then if you still don't know, you kind of test both and look at the output. Right now, because we don't have ambiguous values, we don't have quality values, we don't want to skip any forward or reverse strand of the reference because we don't have, that's not specified, we just have the simple sequences. So under alignment options, we're going to use default values. Next, scoring options. This seems like a lot, but you always have to do this when you're setting up a study to make sure that you're doing the best for your data, the best thing for your data. Once you've used a certain program a lot, like if you use HiSat2 all the time, you'll have a pretty good feeling of what you want to change and what you don't want to change. And so it's time consuming in the beginning, but it's worth it. So specify scoring options. Scoring is usually like when you have a mismatch or, well, there's no ambiguous term, so mostly mismatches. For example, if you have a file, I don't want to click that and click off. If you have, say, let me move this. You have a sequence that's A, and then it maps to the reference with it maps to this reference. This would be a mismatch. So the rest of the sequence, if you can imagine it extending with various characters, so it extends and it matches up all along the extension. It just has this one error. Like, how severe a penalty do you want for that to be? And this error can be something from PCR. It can be an actual difference, like this is a SNP that's present in many individuals, but that's not in the reference file because the reference file is based on like the most common variant, or it might just be an error. It might be an error in the reference, it might be an error in your subject, it might be a real difference that just doesn't matter. So how severely do you want to penalize the mismatch? That is what this is dealing with the scoring. That's usually what scoring is dealing with. So constant term for the above function, I don't know what that means. So if it was necessary, I would go down and read the README and maybe use Google. Coefficient for the above function. By above function, I'm guessing it means, where do we see that? Here, because that's the only other place. This is dealing with ambiguous characters. Whether we need constant, linear, square root, or natural logarithm, I would just write a note somewhere that like we had those options and we selected the default, so that later I can go back and see if that's important. I wouldn't start messing with it at this point. So for the constant term B, coefficient A, I'm going to leave them as default. Maximum mismatch penalty, you can read under here. For now, we're going to leave the default, but we're going to note in our mind or or like on Post-it that this might be something we want to change if we get bad results. Minimum mismatch penalty, we're going to leave that alone for now. So basically, we would just know, we would use the default, so we would use the default for all of this, but if our mapping goes, or alignment, here they mean the same thing, alignment and mapping. If it goes horribly wrong, we're gonna note that scoring options might be something we want to return to. Spliced alignment options. We don't want to deal with splice sites right now. So we are going to leave this as the default. Reporting options, primary alignments, output options. Write unaligned reads in FASTQ format to separate files. Yes, you always want whatever reads were not used, you want those in a file so that you can look at them. If you find that those reads, like if you do a blast on them and you find that they map to the human genome and they map to microRNA sequences, then you're going to want to investigate why they didn't map. So maybe it was one of those parameters we looked at previously that we need to go in and change, or maybe it's a problem with our reference file that's actually missing certain microRNA reads. Contrarily, if you do a blast and you come up with like prokaryotic stuff or genes, actual genes like mRNA, then you'll know that that's okay. It's okay that those were unaligned because those should have been unaligned if we're just looking at microRNAs. And prokaryotic could be like bacteria in the lab. I always think like a lot of stuff gets in the sequencing sample. Like all of the stuff I did with cows probably had a lot of my DNA in there. I would always blast to try and find human DNA. DNA, because after you work with it in the lab, you're working with DNA. That's why your reads to are DNA sequences. It doesn't have uracil, it has T. Because in the lab, you change mRNA to DNA. Very pedestrian. This is very basic. Write aligned reads in FASTQ format to a separate file. I don't know if we're going to need that, but usually I get all the output I can, and sometimes I'll save that somewhere. I don't know why it's doing in a FASTQ format. It's not going to have quality scores, so we can change that to a FASTA format. But for now, it looks like sometimes people enter FASTQ formats into this program. So maybe the default is to preserve the quality scores that are entered, which it doesn't matter for us. Okay, so we're going to leave that. Other options, I don't know what those are, so we have to open it up. Use this number as the seed for pseudo-random number generator. So random number generator, it's called pseudo because in any code, however the random number is generated, it's not really random, right? Like everyone knows random.org, and if you read about random.org, that's built on some sort of atmospheric numbers, and that's believed to be as random as we can get. But in programs, there's always a formula to generate the random numbers, and some programs have better random number generators formulae than others. Java is pretty good. Some, it runs in, that's probably more that I need to get into. Some, the random number is fine for basic tasks, but if you're doing a large data set, you run into problems. So like a true random number generator should have overlaps, and with some, this is like a basic principle of statistics that random numbers, true, that a lot of people don't understand. So there's certain graphs, if you have a true random graph, you basically will end up with overlaps. So these are, you shouldn't have any patterning like this, but you should have overlaps, which I believe would be like the black areas, dark areas, and you should have clusters. That's how random stuff works. If you have something that's just completely looks like static, which is actually what this looks like, but I trust random.org. So like this I would be less inclined to believe that this was a true random experience. You can see here, well, that's something different. Anyway, basically, sometimes you will want to specify a seed for the pseudo-random number generator, or you will want to enter in your own formula. For the seed, I don't know how it would change here, so we aren't going to mess with it. I don't know how seeding would change, because you're still using the same formula, you're just starting at a different point. I would have to read up on what that does, but we're gonna leave it as 0. The default, reinitialize the pseudo-random generator for each read using the current time. So this is to get away what I was trying to explain. This is one of the most basic ways to get away code, quote-unquote, random number generation that runs into a problem, is you use the same formula, but you restart it. And you restart it using, I don't know if this would reseed it on the current time, but you basically have to restart the process. You can read up on this if you want to know more. I don't know off the top of my head. I have it written down somewhere. It seems like the current time would seed it, and by the current time it goes to milliseconds, or like time in milliseconds, which is how computers read time anyways. So if you thought that this was going to be a problem, random number generation, and the way you would think that is if you're like super math nerd and you've worked it out, or if you read a paper where people suggest that it could be a problem, then you would want to play with these. We don't want to play with these right now. I would like to run one with and without this just to see if it makes a difference, but I'm not going to put you through that. I'm really obsessive about like getting the right data, because I figure long after I'm gone and like people find out other answers for diseases and what things are important, if my data is good, then they can go back and use that data to figure out like what genes are important, or to verify that certain genes are important. So I'm really obsessive about getting all of these processes right, and that's probably the best way to learn how to do it. Some people just come in and they're like, yeah, default, that's fine, which is kind of what we're doing in the beginning, but they don't bother reading anything. You always want to read you want to read this, you want to read all this, you want to read the readme, which is all this. Usually monospace type will suggest that there's a readme file. But you want to really know what the program is doing. For now, I'm leading you through everything, so you're just gonna trust what I tell you. But I wanted to walk you through, we're gonna run this now. I wanted to walk you through, so execute, and we got an error. I'll look through that and I'll tell you exactly how to set this up. But this video is important because you need to know that you need to look at the tools you're using and you need to read stuff and try and understand stuff. And if you have a question, if something sounds like it's really important and it might matter, like say the scoring might really matter. And the default is usually built on what is good for most data. And since what we're working with is pretty much like working with human stuff, human or other eukaryotes with a well-defined reference genome, it's pretty standard. That's as standard as you can get versus working with a bacteria where there's no genome, there's repetitive sequences that people, I don't know, maybe there's a lot of plasmid transfer or repetitive sequences that people don't really know the length of. Although now with long read generation, I hesitate to say that because now that problem is kind of solvable for prokaryotes, not for all eukaryotes. But if you find something that you think is important and you just don't know, is this value right? Two ways to approach that: test different values. So test 6, then test 0, then test 20, and see how it changes things. It might change them a lot, it might not. And you want to go through anything you think is important after reading about it. You want to go through and test it for yourself, and then you also just want to look and see what other people, so you would go to Google. I'm going to copy this. Go to Google, type in the software HiSET2 maximum mismatch penalty, and then you'll see stuff from BioStars, from Seek Answers, from, don't ever use ResearchGate. This is like the dumbest of the dumb get together and ask dumb questions. So stay off ResearchGate. Unless you have to create a profile at some point, just avoid it. It's really stupid people. This is their actual paper, so you might want to read that. It might be a little dry and hard to understand if you're starting out in bioinformatics, but you look for pages with BioStars, Seek Answers, or Stack Overflow, and you just read. This is people asking questions and other people answering it. And they'll probably have the same questions that you have, and you can kind of get a better idea of what the tool is and whether it's important, but you still want to test it yourself. Also, when using these, check the dates. So some of them 2014. The principles might still be relevant, but the software, this is Bowtie 2, so it's not even HiSat, but the software may have changed drastically in intervening years. So, yeah, those are the two approaches when you think something might be important, you just aren't sure. Test it yourself and go and read other questions. Whatever tool you use, if you go into bioinformatics, you need to study the tool. Most people don't do that, and you can't trust their data. And for neurodegenerative diseases especially, it's really bad because bad information is put out there, and some people build their whole career research on this bad information. It sets us back a lot in terms of figuring out diseases. So I'm going to figuring out diseases, more or less. I'm going to figure out what parameters we need, why that threw an error, and I'll send you the exact parameters, or I'll make another video of them. Basically, you would run execute, and then it would go over here, and it would turn from a light gray to a yellow, which means it's running. If there's a failure at some point, it'll turn red. And for you just ask me, it turned red, what do I do? If it's successful, it'll turn green, then you move on to the next step.